Microfluidics refers to the study and manipulation of fluids at the microscale, typically in channels that are mere micrometers in width. These channels allow scientists to control small volumes of liquids with extreme precision, leading to more efficient reactions, faster processes, and the ability to run numerous experiments simultaneously. One of the key applications of microfluidics is the lab on a chip, a compact device that performs complex laboratory tasks such as chemical reactions, mixing, and analysis, but on a chip the size of a postage stamp. This technology is especially valuable in fields like diagnostics, drug discovery, and personalised medicine, where time and efficiency are of the essence.
Think of microfluidics like running a full-blown chemistry experiment inside a dollhouse kitchen. You've got everything you need, but in tiny amounts. Imagine Gordon Ramsay trying to cook a 5-course meal, but instead of a full kitchen, he's working on a stove the size of a quarter. He's sautéing, boiling, and plating, all with an eyedropper and a speck of salt. That's microfluidics, compact, controlled, and highly efficient.
At the microscale, the physics of fluid behaviour changes drastically compared to what we see in larger systems. In everyday life, we often observe turbulent flow, where liquids swirl, mix, and create chaotic patterns. Think of a river or even cream mixing into coffee. But at the microscale, laminar flow dominates. This means the liquid flows in straight, parallel layers with minimal mixing unless intentionally forced. Surface tension, which refers to the cohesive forces between liquid molecules, becomes a major player. It can cause fluids to form stable droplets, resist breaking apart, and behave differently in small channels than they would in a larger container.
Imagine a world where rivers flow like they've taken a crash course in etiquette. No wild splashes or chaotic mixing, just smooth, orderly movement, like ballerinas gliding in perfect formation. And surface tension? Well, that's like the clingiest friend at a party who insists on holding hands with everyone in the group. They're inseparable, and they keep the liquid in line too.
Mixing fluids at the microscale presents a significant challenge because of laminar flow. Liquids don't naturally mix as they do at larger scales. In turbulent flow, liquids swirl and blend on their own, but in microfluidic systems, they stay in their lanes unless a force is applied. Scientists use techniques like chaotic advection, where the design of the channels causes controlled swirls and eddies, forcing the liquids to mix. Another mechanism is diffusion, where molecules move from areas of high concentration to areas of low concentration, but this process is slower. By carefully designing the microfluidic channels, scientists can ensure efficient mixing even in these tiny systems.
Mixing in microfluidics is like trying to stir your soup with a toothpick inside a thimble. It's a delicate, slow process. But scientists are clever. They design the channels like tiny water slides, adding twists, turns, and bumps to make sure the liquids swirl together. It's like inviting your friends to a dance party, but instead of just standing around, you've rigged the floor to make everyone bump into each other until they're finally mixed up.
In microfluidic devices, chemical reactions occur faster and more efficiently because the reactants are confined to small volumes. The increased surface area to volume ratio means molecules have more opportunities to collide and react, speeding up the process. Additionally, only minute amounts of reagents are required, reducing waste and making the process highly cost-effective. This is particularly useful in applications like drug discovery, where large numbers of experiments must be run quickly and with minimal material.
Picture a science fair, but instead of full-sized volcano models and beakers of bubbling liquids, every experiment takes place in a matchbox. You only need a drop of this and a sprinkle of that, and voila, reaction. It's like cooking a meal in a dollhouse kitchen with a tiny spatula, except every dish cooks in seconds and uses the smallest possible amount of ingredients. Efficient, fast, and honestly kind of adorable.
Microfluidics has become a key tool in biological research, particularly for manipulating cells. With microfluidic devices, scientists can control the flow of fluids to isolate and sort cells based on specific characteristics. This technology is invaluable in fields like cancer research, where circulating tumour cells, CTCs, can be separated from a blood sample for analysis, helping with early detection and treatment monitoring. Microfluidics allows for precise manipulation of cells with minimal amounts of fluid, making it a highly efficient process compared to traditional cell sorting techniques.
Microfluidics is like a high-tech bouncer at the world's tiniest nightclub for cells. It checks each cell's credentials as they arrive. Some are VIP tumor cells, some are regulars, and some are turned away at the door. No pushing or shoving here. Everyone gets neatly sorted and goes exactly where they're supposed to. It's like watching a microscopic version of a TSA line at the airport, where cells are scanned, sorted, and sent on their way.
Microfluidics has revolutionized point-of-care diagnostics by making it possible to run complex tests on-site with minimal equipment. Using just a few drops of blood or other bodily fluids, microfluidic devices can test for infections, measure glucose levels, and even detect diseases like COVID-19. These devices are portable, fast, and highly accurate, providing results in minutes instead of hours or days. By shrinking the entire laboratory process onto a chip, healthcare providers can deliver immediate, personalised care without needing a full lab setup.
Think of it as having a personal lab assistant that lives in your pocket. Got a cold? No problem. Drop a bit of fluid into the device and wait for it to tell you what's up, like having a doctor who texts you the results in real time. It's the fast food drive-through of medical testing, except way more accurate, and no need to wait for weeks in a lab queue.
Microfluidic technology allows for precise and controlled drug delivery, especially important in targeted therapies like cancer treatment. By embedding drugs into microfluidic channels, scientists can direct medications to specific areas of the body, reducing side effects and increasing treatment efficiency. This targeted approach ensures that only the affected cells, like cancerous tumours, receive the medication, leaving healthy cells largely unaffected.
Imagine you're in a garden with one particularly needy plant. Instead of watering the whole plot and wasting water, you use a tiny squirt gun to spray just that one plant, no more, no less. That's how microfluidic drug delivery works, ensuring the medicine hits just the right spot. It's like being a molecular sniper with perfect aim.
Microfluidics is playing a crucial role in environmental monitoring and water purification. Tiny filters embedded in microfluidic devices can remove pollutants from water, while sensors within the system detect contaminants at incredibly low concentrations. These devices are portable, making them ideal for real-time testing in the field, where immediate results are essential for ensuring water safety.
Picture a tiny Brita filter strapped to your backpack, purifying water one droplet at a time while you're hiking through the woods. Or imagine a microscopic robot fish swimming around in a lake, gobbling up contaminants like they're snacks at a buffet. That's microfluidics in environmental science, cleaning up the world's water supply one drop at a time.
Microfluidic systems offer tremendous advantages, including faster reactions due to the smaller volumes involved, increased efficiency because of reduced reagent use and greater precision in controlling how fluids mix, react, and move. These benefits make microfluidics ideal for high-throughput experimentation, diagnostics, and drug development, where time, cost, and accuracy are critical.
Microfluidics is like hosting the world's fastest cooking competition. You give chefs tiny ingredients in just a few minutes, but somehow they pull off a gourmet meal. No waste, no mess, and every dish is perfect. It's the molecular version of speed dating, except the only sparks flying are between reactants in a microchannel.
Despite its potential, microfluidics still faces challenges, particularly in the manufacturing process. Creating devices with such tiny, complex channels is costly and requires high precision. Scaling up these systems for industrial or widespread medical use is another hurdle. However, the future of microfluidics looks promising, especially with the integration of artificial intelligence and automation. AI could make these systems smarter, allowing real-time adjustments to processes based on the data collected, leading to more personalised healthcare and more efficient industrial applications.
Imagine trying to build a miniature amusement park inside a snow globe. Every ride has to be tiny, perfect, and functional. That's the challenge of making microfluidic devices. But once we figure it out, the future is bright. Picture an army of tiny robots in lab coats, running experiments inside your body, fixing problems before you even know they're there. It's like having an entire team of microscopic MacGyvers keeping you healthy.